You guys met, right?
Yeah.
Yes, Hari.
So I will do a small brief introduction.
And then
Good go my intro. Board this and ಎಲ್ಲರೂ ಹ್ಯಾಂಗ ಕ್ಯಾಟ್ ಹಾಕೋಪ್ರ ಇಪ್ಪ So he was from Chennai, right? So.
this, we had a lot of connect.
Okay. Oh, you also had some.
He has relatives in Chennai.
Okay. ಇಲ್ಲ என்ன ಕರೆಕ್ಟ್ anyway. All right. I take it. So thank you all for coming to this seminar session here. So today we have Professor Anand Mohan. He's a professor in the chemical engineering department at IIT Hyderabad. So I'll just give a brief introduction what is pedigree here. So he received his BTech in chemical engineering from IIT Madras in 1999. He got his PhD from Texas A&M in 2005. So primarily his PhD research resulted in a unified framework of models to study blood flow accompanied by clot growth and lysis. So after that he worked for 4 years in the oil and gas industry at CGGV Veritas, Houston. And after that he joined as an assistant professor at IIT Hyderabad. So now he's full tenure faculty and full professor from June 2024 as a full-time professor. So he has a lot of papers, and I think today he's going to be talking to us about diagnosis of cardiovascular diseases using model simulations. So some small introduction, please.
Thank you for the introduction, Karthik. I first thank IIT Gandhinagar Department of Chemical Engineering for covering my local hospitality. Thank you very much. I thank DBT India for covering my travel from Hyderabad to Ahmedabad. This talk is part of outreach for my DBT funded project. So thank you for that. And the talk is titled Diagnosis of Cardiovascular Diseases Using Model Simulations. The outline is as follows. First I will motivate the study of the problem, give some historical remarks ending with what is known as Workhoff's triad, then talk about the problem setting, the problem formulation, then give the results for 2 problems that we will study in this talk, summarize the results and conclude with my acknowledgements. Okay, so cardiovascular diseases are the leading cause of mortality, whether in the world at 33% or in India at 29.5%. And the underlying phenomenon of cardiovascular diseases is blood flow with accompanying clot formation or lysis. So if we want to understand cardiovascular diseases, we need to understand blood flow with clot formation and lysis. Okay, so the flow is not just like flow through of a water through a pipe. The flow is combined to biophysical processes like diffusion, permeation, cell migration, etc. And there are literally hundreds of individual biochemical reactions underlying clot formation. For example, thrombin activation of fibrinogen. Now, with modeling, that is with rheological models of blood and clot, you can predict velocity and shear stress patterns inside the arteries. With mathematical models of coagulation and fibrinolysis, we can generate hypotheses. Now, the question that I will be asking in this talk is, can they assist in diagnosing CVDs and interpreting laboratory data? Okay, and so some historical remarks on the study of blood flow. The study of blood flow goes back to the ancient Egyptians. Okay, they had a very good knowledge of the inner constitution of the human body, saying that the heart had the central role and there are 46 blood vessels to various parts of the body. Greek civilization, which followed next, revered the knowledge of the medical knowledge of Egyptian civilization. But the central figure in Greek civilization is the person known as Hippocrates, who separated the practice of medicine from the practice of religion. That means that a disease is due to some natural cause and not because you had offended some god of rain or god of water, things like that. Okay, so he is the central figure and in the Greek civilization, and he looked at disease as occurring because of inequilibrium of 4 humors in the human body. Similar theories were there in the ancient world. In Ayurveda, you have the 3 doshas that are recorded in the Charaka Samhita as Vata, Pitta and Kapha. And you have in the inner canon of Wang Di of Chinese medicine 2 principles, yin and yang, which interact for the maintenance of health. Okay. So the next part of this journey of blood flow occurs due to in the Roman Empire, where there is a Greek physician named Galen who refined Hippocrates' theories and advocated bloodletting as a cure for diseases. For several centuries afterward, bloodletting was the go-to cure for any sort of ailment in Europe. Okay. This changed when modern studies in clotting took place with the discovery of red blood cells by Leeuwenhoek in 1674 and the discovery of platelets by Alfred Donné in 1842. Richard Weissman was an English surgeon who said there are 2 causes for thrombosis. Thrombosis is excess clot formation. Okay. But the modern understanding of thrombosis comes from the German pathologist Rudolf Virchow, who identified 3 causes for thrombosis in 1856 and which is now called Virchow's triad. So what is this Virchow's triad? Okay. Virchow's triad says that thrombosis is due to either hypercoagulability, that is something inherent in the blood for leading to clot formation, or flow stasis, that is some change in the flow pattern, or an endothelial injury, something affected at the surface of blood flow when it flows through the arteries of the human body. Okay. So this is the bedrock of our understanding today. So how do we see it today? Virchow's triad today says that blood fluidity is held in a delicate balance due to interactions between blood flow and surface. Okay. If there is a balance of these 3 legs, there is no clot formation. If there is an imbalance in any leg, like for example in the surface you say that there is an injury, then you have clotting or coagulation occurring. If the imbalance is removed, that is after the underlying injury heals, then the clot lysis happens and the blood fluidity is restored. So notice that here when we say Virchow's triad, the Virchow's triad in 1856 talked about only thrombosis, but now we talk about it as affecting clot formation per se. Thrombosis is excessive clot formation, but it is now understood for clot formation itself. If there is a problem in this balancing mechanism, then you have a clotting pathology. Okay. So let's look at the setting in which blood flow with clot formation takes place. Okay. So you have the heart as the central pump, and it pumps a fluid tissue called blood through a network of arteries, arterioles, capillaries, venules and veins back to the heart. And its primary function is the transport of oxygen, nutrients and waste to and from the various parts of the body. Okay, so let's look at blood, okay, and its composition and its rheology. Okay, so blood consists of formed elements of 46% by volume in aqueous plasma. And these formed elements are primarily RBCs, which constitute 98% of the formed elements, and they are viscoelastic. The remaining is platelets, which is also viscoelastic, and some white blood cells, which goes up during disease, which is less than 1%, also viscoelastic. Now these viscoelastic formed elements are suspended in plasma, which is an aqueous solution of proteins in water. Plasma is Newtonian, and the overall mixture, as you expect, exhibits complex rheology. Blood is a shear thinning, viscoelastic, even thixotropic fluid, but I will be focusing only on blood as a shear thinning fluid in this talk. And the clot is a very viscous, viscoelastic fluid, and I will be focusing only on its very viscous nature in this talk. Okay, so this is blood rheology. Okay, now let's see what happens when the clot forms. Okay, so you can see here, I told you that if there is a rupture in the injury, rupture due to injury, you have all the blood cells rushing to the site of the injury, and by the chemical reactions of coagulation, a network of polymeric strands known as fibrin is formed, which traps the RBCs and platelets inside a network. Okay, here this same image gives you a picture of what is happening. Okay, so this is what we are trying to study of clot formation in blood flow. Okay, so we are ready to pose the problem, okay, that blood flow with clot formation and lysis involves the rheology of complex fluids. I told you blood has a complex fluid rheology of blood and clot. The biochemistry of reactions involved, that is coagulation and fibrinolysis, and also the fluid structure interactions between blood and the vessel walls. This I will neglect in this talk. I will focus only on these two parts. Okay. Now I will look at two problems. That is, one is the simulation of blood flow in a rigid walled abdominal aorta. That is, the novelty there is that we are studying a two-phase fluid in a patient-derived geometry. And we'll also look at blood flow with clotting and lysis in what is known as thromboelastography, which is a diagnostic device. Okay, so here I will have a nonlinear fluid combined with reaction ODEs. Okay, so the hypothesis that I am making is that model simulations yield hypothesis and workflows in cardiovascular mechanics, and these can be used to diagnose CVDs. Okay, so the first problem. Okay. So when we look at the literature in blood flow in rigid walled geometries, okay, we have the literature split into two parts. The literature consisting of idealized geometries and the literature in patient-derived geometries. If we look at the idealized geometries, we get results for very complex fluid models. Shear thinning viscoelastic fluid, mixture theory model in straight pipe or rectangular channel. But when we go look at the work in patient-derived geometries, we see only much simpler rheological models, Newtonian fluid or Caro fluid, shear thinning fluid. Now the novelty that we have achieved is to look at a complex mixture theory model in a patient abdominal aorta, and this was due to my student Amin and I, who published in the Journal of Biomechanical Engineering. Okay, so the CFD workflow to predict sites of atherosclerosis was the primary result from that paper that I referred. And see, first we have a geometry which is derived from patients. Okay, so we have DICOM images from CTA scans. Okay, and we extract from the DICOM images a segment of the abdominal aorta. Okay, and once that abdominal aorta is obtained as a geometry using the SimVascular software, we import it into ISM CFD and do the meshing. And we have polyhedral cells, as you can see here. And the cell count is determined based on grid independence criterion. Okay, and this is for a velocity in the midsection. Okay, now that we have defined the flow domain and we want to simulate the blood flow, I told you we will look at a mixture theory model. So what is this model? Okay, so we have a constitutive model for blood, which is a two-phase fluid mixture of a volume fraction weighted Navier-Stokes fluid, that is plasma, and also a volume fraction weighted shear thinning model for the RBCs. Okay, so here you can see the RBC model has a viscosity which is dependent on the volume fraction and the shear rate. Okay, and the net mixture has a viscosity that is given by this volume fraction weighted viscosity of the fluid plus the volume fraction weighted by the solid, viscosity of the solid, and we have a mixture that is given by this. And this mixture viscosity fits the experimental data in Brooks et al. This model we published in a paper in Aminuddin et al. 2019. Okay, so this is, now we have defined the domain. we have now defined the constitutive model. Now let's see what are the balance equations that we have to apply, substitute them into. Okay. So the governing equations are of mass balance, which is what you will call as the continuity equation. And then you have the governing equation for momentum balance, but they are written for two phases. One for the plasma, one for the RBCs. Okay. And you have an interaction force here. This interaction force consists of terms from the density gradient, from the drag force, and also from the lift force. Okay. We could get more complicated, more fancy, but this is what we have done at this stage. Okay. So the drag force, you can see, if the solid moves faster than the fluid, this will become positive, and its net effect will be to retard the motion of the RBCs. So it is physically consistent. Okay. So we substitute these, the constitutive model in the flow equations, and we calculate hemodynamic indicators that are known to predispose to atherosclerosis. This is from another paper. Okay. And the hemodynamic indicators are total average wall shear stress, oscillatory shear index, and also relative residence time. And along with these hemodynamic indicators for atherosclerosis, which are calculated during the flow, we also simulate the concentration of low density lipoprotein. Okay. And through a convection diffusion equation. Okay. And the numerical scheme is in ANSYS Fluent, which is an Eulerian model with a second order upwind scheme. And we ensure grid independence before I present the results to you. We compared the maximum and minimum velocities at the midsection and made sure that the percentage difference is less than 2%. Okay. So what does the complete CFD workflow give us? Okay. So here you can see in this abdominal aorta, based on the total average wall shear stress, you have a large patch forming here, which represents the possible location of atherosclerotic plaque. This large patch is refined by the other hemodynamic parameter, the relative residence time, in this picture. And when we see the concentration of LDL deposited on the wall, the patch is much smaller. So what this says is, if we take the time and effort to calculate the concentration of LDL, low density lipoprotein, then we can much more accurately predict the location, site and extent of the atherosclerotic plaque. Okay. So this is the first problem that we studied. Okay. So the second problem is of thromboelastography, and this involves blood flow with coagulation in an oscillating cup. I looked at only blood flow in a rigid walled abdominal aorta in the previous slide, in the previous problem. Now I will look at it with blood flow with coagulation, but my walls will be rigid. Okay. So the conventional laboratory tests are performed called prothrombin time or activated partial thromboplastin time, and they are performed on what is known as platelet poor plasma. Now because they remove the platelets from the plasma while taking these tests, it cannot capture the true rate of clot formation or the clot strength. But these are important, and they are very valuable to assess the hemostatic status of a patient's hemostatic system in, for example, liver transplant surgery or during cardiac surgery. And the new diagnostic method that is used is called thromboelastography, which includes the elastic properties of clot, and it has a quick turnaround time. It can be used at the bedside itself. Okay. And thus far, we have only one ODE model for TEG that has been developed, whereas what we propose to do is to come up with a comprehensive model that combines both mechanics and biochemistry. Okay. So before we go there, let's see what is TEG. You have you have a little cup of blood here. Okay, this is from a personal communication that I got this picture of this blood sample. You keep this blood sample in this cup here, and the cup then rotates. Okay, and if the cup is oscillated, then you have what is known as thromboelastograph. If the pin, on the other hand, that is suspending here oscillates, then you have rotational thromboelastometry. Or if the probe oscillates in and out of the cup of blood as it is clotting, then you have sonoclot. Okay, the TEG output will give you the rate of fibrin polymerization and the overall clot strength. So how does this actually work? Okay, let's see that in the next slide. Okay, so you have a blood sample that is drawn by phlebotomy. It's placed in a cylindrical cup, and as the blood in the cup starts clotting, you start rotating the cup. Okay, then you place a pin with a torsion wire attached to it on top of this blood, and as the blood clots, the torque will be changed, and that will be detected by an electromagnetic transducer, which will create an electric signal and then a graphical output, and the shape of that graphical output is used to determine the state of a patient's hemostatic system. Okay, so how does this work? Again, I have a video for that. Okay, so you have this cup which is oscillating. The pin is now brought and kept on top of it. Blood is starting to clot, but you can see the tracing that has come, and as the clot starts to occur, the tracing starts to increase in amplitude. Okay, so this will happen as long as more and more blood clots, and once that clot breaks after a certain period of time, because you don't have any continual refilling of the enzymes that are being consumed. Okay, so the clot starts to break and the amplitude of this decreases. Okay, so this is how a normal TEG tracing looks like. Okay. Okay, I'll stop here. Okay, I think this gives you the idea. This will go until the TEG tracing becomes back to 0 again. Okay. Okay, so the first model of TEG, and probably the only model that we could find in our literature, is that of 4 species: platelets, activated platelets, thrombus state, and lysed thrombus. And there are 4 ODEs. The authors plotted the graph for T of T versus time and fitted these parameters such that the T of T tracing matches what is recorded in the TEG tracing. Okay, now this is not sufficient. This is not a very crude way of modeling what is happening in the TEG. Okay, so we came up with a new model for TEG by Mridula and in her MTech thesis along with co-workers. We obtained the flow equations of blood in a cup using what is known as the semi-inverse approach. We then extended the shear thinning model for blood developed by Yeleswarupu et al., where you have the shear stress with the viscosity dependent on the shear rate and the concentration of fibrinogen. And how do we combine this with biochemistry? See here, as far as I am concerned, I have only studied a mechanics problem. How do I combine it with the biochemistry? It is by taking this I from the equations that I already told you of Presley et al. So I combine it with the 4 ODEs for clotting given before, and I set the thrombus state in the ODEs to the fibrinogen that is there in this model equations. Okay, so now when I calculate the displacement, calculate the velocities, and from then the displacement of the pin, I can get my TEG tracing. This TEG tracing we validated with data from healthy human males, and then we predicted it for a multiparous female, that is with or without platelet deficiency. And you can see that picture in the next slide. So here you can see the model is giving reasonable fit to the data, and here it is much better. And the good thing about this model is that it is increasing its amplitude with increasing platelet count. Okay, so that much level of qualitative match comes out from this model itself. Okay, and so to summarize, mathematical models and computational tools yield hypotheses and workflows in cardiovascular mechanics. We proved that these can be used to diagnose CVDs because I showed you one CFD workflow to predict the sites of atherosclerosis. I also showed you a unique interdisciplinary model to predict the tech tracing by Mridula and co-workers. And so in conclusion, what I hope to persuade you to consider is that mathematical models and computational simulations can assist doctors in diagnosis and decision making, assist in the interpretation of laboratory data, and it has the advantages that it is non-invasive and cost-effective. Okay, so I will conclude with my, there is future work that I would like to do that we plan to take ahead. One is the simulation of blood flow with clotting. Okay, so I take the CFD workflow, I simulate it with a multi-species mathematical model which by Susri et al., which includes the latest platelet activation and binding mechanisms. The other project that we are currently working on, and this is the funding for this DBT, for the project that covered my travel, is that of simulation of blood flow using an efficient framework where we will parallelize the sequential code for blood flow in a stenostomy tube. And so we hope to come up with a much faster way to solve these flow problems in real time. Okay, because those pictures that I showed you took like 132 hours of computational time. So it needs much faster time response to make it attractive for real-time diagnosis. Okay, so that's what this DBT project is about. So with that, I conclude with my acknowledgements. I thank the real workers, Aminuddin, Mridula, and Sudarshan. My collaborators, Professor Masoodi of CMU, Professor Antaki of Cornell. I thank the constructive feedback from my colleagues Vinod Janardhanan and Renu John and IITH. I also thank the funding agencies, Ministry of Education for the IITH students and the DBT project with project reference 41173 for this travel grant. Thank you so much for your time. I'm ready for your questions.
A lot of information about blood flow that you might not have learned before. So before going further, questions from the audience and students. Any questions?
Yes.
Two questions. So one, when you show that TEG kind of a measurement, is it analogous to the oscillatory, large oscillatory measurements we can do in the rheometer?
It is, the principle is the same. It is just like a large amplitude oscillation. But the important thing is what is the torque transducer measuring and how is it converting it into a graphical output. That is what is the analog stops with just where you do the torque transducer. But do you have a way of taking the torque transducer and converting it into a graphical output? That part is still, how the transducer converts it into a graphical output is still not understood. That we are hoping to address with this model.
Okay, but I'm not clear. What is the technique measuring? Or maybe I missed it. What is it measuring?
It is measuring. Okay, just 1 minute. See, the cup oscillates,
yes,
and the torque is what is measured. Torque is measured. So in that sense, it is analogous to a large amplitude oscillatory shear. But how that torque is converted into a tracing is still done by the transducer, and that doesn't come under this. So then I have to go into how it's converted into an electric signal and how the tracing is converted. That part is still black box for me in this model.
Okay. And in the last slide, you showed those two graphs, right, with the platelet count. Is it platelets?
Yeah, this is with platelet count 90,000 per microliter. This is with platelet count of 233,000 per microliter.
So you said one, it does better than the other.
Right.
So my question is, to me, I mean, what should be my criteria for predicting better? Is it the final stage I am looking for, or the initial linearity which you have shown in the
See, from
because
this,
in here it is somewhat closer to the linear portion, and then your points are off in the
curved portion.
right,
Correct.
constant, but
Correct.
the other, it's perhaps better in the constant portion
But-
and it's variating in the linear portion.
Correct,
right?
correct.
So what is the criteria, like,
See, that's why I stopped it with qualitatively better, saying quantitatively this is good.
Okay, I'm done.
Okay.
Yeah.
Ideally, you should get it to go through every point.
Every point, yeah.
But you will not, I mean, it is difficult to get a model to do that, okay? Because this model is trying to capture what is happening in that black box between the transducer and the tracing, okay? So, I mean, I can say here that this looked, I mean, see, the extent of misfit here is more,
Yes.
whereas the extent of misfit here is less,
Yes, yes.
okay? Ideally, this part is the part which you want to correlate much better,
Okay,
because that relates to the rate at which the clot is forming.
I see.
Okay, this is the rate of fibrin polymerization occurring. Here it is something like the maximum strength of the clot. It's okay if you are off in the final strength of the clot, but the rate of fibrin polymerization determines how quick your hemostatic system is going to respond during a surgery and how his injuries are going to get healed. So I would say this is the part that we need to see if we want to see how good the match is. Okay?
Thank you.
Thank you. Thank you. Very good questions. Thank you so much.
So I just have one small question.
Yes.
So again, the same data here. The initial time points do not seem to be captured. Is there a specific reason for that?
It's a good question, but I have not thought about it. I have not thought about why it doesn't capture the initial few data points where, say, because our mechanistic calculations, assume that the measurement can be made from time zero itself. But from time zero, when the cup is placed on the torque transducer, we don't know how much time it took for the clotting to start happening. So that part, I am assuming that immediately my equations will start solving the equation and I will get a displacement. But that is, that part is being missed in this initial part, when the clot is kept inside the cup itself.
So I just had a question where you are measuring this and you are applying torque, and sometimes the cup is also rotating.
Yes.
So this affects the kinetics or of even the clotting itself, right? So how do we...
No, the kinetics of the clotting is irrespective of how much how the torque is going to affect. See, because the torque transducer is just placed on the surface and the clotting enzymes are being consumed within the cup of the blood itself.
So if I rotate the cup, for example,
Correct.
if I have blood and let's say RBC, then all these cells are there.
Right.
So if I rotate it sufficiently fast, then because of the centrifugal force, there is a tendency for the cells or the RBCs to come to the surface.
Yes, but these instruments are standardized for some rotation speed. I told you that the tech is standardized for 4 degrees 45 minutes in 5 seconds. So that is standard. You cannot change. I will say I will rotate it at much faster and see if my tracing will be difficult. Because this standard has to be applied for this instrument for it to be used in labs around the world. So this standardization is kept with the idea that I don't want to change the strength of my clot by changing the clot speed too much.
If you have no further questions, then let's just thank the speaker one more time for your time.
Thank you all for coming.
Thank you. I think there's snacks outside as well, so that's all we kept. Please help yourself. So after that, please go and interact with the speaker. Please do come back. So first, let's